MatTek's EpiDerm Full Thickness 400 kit. EpiDerm Full Thickness 400 kits are available for shipment every other Monday. They are shipped at 4 degrees Celsius and delivered Tuesday, Wednesday, or Thursday, depending on the receiving location. Refer to the use protocol provided or visit our website EpiDermFT page for more information. Upon receiving your shipment, remove all kit components from the box. Each kit contains 1 EpiDerm Full Thickness 400 use protocol, 24 ready-to-use EpiDerm Full Thickness tissues in 4 sealed 6-well plates, 4 sterile 6-well plates, 1 100 milliliter bottle of PBS rinse solution, 1 10 milliliter conical tube of 1% Triton X100 solution as an optional test control, and 1 125 milliliter bottle of assay medium. Place both the EpiDerm Full Thickness tissue plates and the assay medium in the refrigerator, maintaining between 2 and 8 degrees Celsius until you are ready to use them. Once you are ready to start your experiment, preferably the same day of receipt, tissues should be returned to culture. To do so, using sterile technique, place the assay medium bottle, the 4 6-well plates, and the 4 EFT-400 tissue plates provided in a tissue culture hood. Remove plastic wrapping from each of the 6-well plates and label each plate accordingly as you see fit. Next, carefully remove the plastic seal from the assay medium bottle and, using either a serological pipette or auto repeater, dispense 2.5 milliliters of assay medium into each well of each plate. Gently swirl each plate to ensure the surface of each well is covered evenly with the assay medium. Taking hold of the EFT-400 tissue plates, using either scissors or scalpel, remove both layers of packaging and carefully slice the lab tape where the plate cover meets the base. Then, with sterile forceps, remove the gauze and place on the cover. This can be used to remove excess agarose from the insert. Pick up the plate to visually inspect the tissues. With the sterile forceps pinching the edge of the insert, Pull it out of the agarose, being careful not to puncture the tissue, and place in one well of the 6-well media-filled plate. Once all tissues have been transferred, place the plates in an incubator at 37 degrees Celsius, 5% CO2, and 95% relative humidity for overnight equilibration, approximately 16 to 18 hours. 10 to 15 minutes before the end of the equilibration period, pre-warm the assay medium to 37 degrees Celsius. At the end of the equilibration period, aspirate the media from each well of the 6-well plates containing the tissue inserts and replace with fresh pre-warmed assay medium. Tissue equilibration is now complete, and your epiderm full-thickness tissues are ready for your experiment. We are MatTek, a BICO company, here to support your science every step of the way. For questions, contact us at 508-881-6771 or email us at support@mattek.com.